The overall goal of this experiment is to demonstrate the detection of microorganisms at very low levels using digital holographic microscopy. This technique is more sensitive than traditional light microscopy and provides real-time information about bacterial behaviors.
This method can help answer key questions in the biological and cosmological fields, such as searching for pathogenic organisms in water or life within our solar system on icy moons.
The main advantage of this technique is that DHM is a volumetric technique, which means we can capture three-dimensional information on our sample instantaneously without any need to physically refocus.
The implications of this technique extend toward the diagnosis of bloodstream or cerebrospinal fluid infections because of the ability to detect low bacterial concentrations.
To begin this procedure, on the day of the experiment, take a spectrophotometric reading of the bacterial master culture, which is expected to be in the range of 0.6 to 0.7. Then, take a sample of the master culture and count the cells directly using a Petroff-Hauser counting chamber to enumerate both live and dead cells. Transfer a 10 microliter sample of the undiluted culture with a micropipette to the chamber. Image it under a high-dry objective microscope using phase contrast. Subsequently, count the bacteria in at least 20 squares and average them. Calculate the concentration as the average of 20 squares times the dilution factor. To enumerate only live cells, make a serial dilution of each of the selected bacterial samples with sterile 0.9% saline solution by transferring 20 microliters of the bacterial solution to another well and diluting it with 180 microliters of saline. Repeat the procedure.